This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 987, recorded on February 24, 2023. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Hi, everybody. All good here. 57 degrees, cloudy.
Does it go from 80 to 57 like in a week?
Oh, yeah. Up and down and up and down. It's going to be 80 again in a couple. Let me see here. Tomorrow's 68. Sunday's 80. Monday's 80. I think we've probably seen our last freeze.
All right. We'll see. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It is a blustery winter day here in Western Mass. We've got ice still left on the ground from the sleet storm, and it's now 29 Fahrenheit. It was in the 30s. It was low 30s earlier in the day, so the temperature's plummeting, and wind is blasting through at 30 to 40 knots. Lovely weather.
We have a guest for you today from Johns Hopkins Bloomberg School of Public Health, Arturo Casadevall. Welcome back.
Thank you for having me back. It's an honor to be here with you.
You were last here in April of 2021. Time flies, doesn't it?
A lot has happened since then, Vincent.
Yes.
There's the understatement of the year.
2021, yeah. A lot has happened since then. And you've been on some of my other podcasts as well, so we're always happy to have you and chat with you. So today we have a couple of topics, including convalescent plasma, because a few months ago we made some statements that perhaps were not correct, and so you're here to correct us. Before we do that, I just want to remind everyone, ASV 2023 registration's open. It'll be in Athens, Georgia, June 24th through the 28th. You can go to asv.org/ASV2023 for information. Members can apply for support for dependent care during the meeting. It's called ASV Cares. Or you can sign up on the meeting site for the on-site child care through KittyCore. You go to asv.org/ASV2023/childcare. So you're in Baltimore. How's things in Baltimore, Arturo? Everything good?
Yeah, it began very warm, and now it's getting colder. We're getting a lot of, by the end of the day, we're going to need a jacket again.
Arturo, there are two jobs openings that I'm thinking that you might be a good candidate for. One is Tony Fauci's job, and one is NIH director. Are you interested in any of those?
No, I think I'm very happy where I am right now, focusing on science.
You would be great at both of those, you know.
You're very kind, but I think it's better if I focus on science.
Now, wait in a year, we'll see he's got one of those jobs, guys. What do you think?
Yeah. I can see him running NIAID.
I have not applied.
Okay. Yeah. All right. All right. So convalescent plasma. Let's talk about that first. You sent us a couple of interesting papers. Just tell us first, what is convalescent plasma, especially for COVID?
So it's simple. It is plasma. Plasma is the liquid part of your blood. If you take your blood, for example, you put it in a centrifuge, you'll bring down the cells. The liquid is the plasma. And convalescent is just what it means. It means you have recovered from it. And what makes it different from regular plasma is that you would have antibodies to whatever you recover from.
Mm-hmm.
And I just want to say that thank you for having me here, and thank you for being so responsive. Neil Greenspan wrote me about the discussion you had about plasma, and I must say that it's wonderful dealing with scientists. Send you a note, and you guys very rapidly got back to me and said, let's talk about it.
Well, if we make mistakes, we want to fix them because we're supposed to be giving people the correct science, right? So we say we're wrong all the time.
So what has been your experience during the pandemic with the whole convalescent plasma story?
So, Rich, it's been all-consuming. I continue to spend 3 to 4 hours a day in this area. This is not what I do for a living. I have a day job. I do other things. But it's also been one of the most rewarding things that I have ever done. Back in March 2020, we went from using 0 units of convalescent plasma to using as many as 25,000 per week by the fall.
Define a unit.
A unit is a 250 milliliters of plasma.
Okay.
And the way that it's obtained now is if you're going to donate it, they sit you down, they put you in a machine called a plasmapheresis machine. And it basically just takes out the liquid and leaves your red cells behind. But any of us can give 2 to 3 units in a sitting.
Okay.
Now,
So-
this is something we talked about early in the pandemic, and this is not some new idea, as people have probably figured out just from the description. It is a very simple thing that's been done in medicine for a century plus, right? And
First Nobel Prize.
right.
Let's give it
is that
to
right?
Emil von Behring. So yeah, you talking about taking something out of the cobwebs. You took out the first effective therapy, but the amazing thing is that it's always there first. So if we get hit with bird flu, you can be sure that it will be rolled out again.
And
Now-
people have kind of vacillated about the effectiveness of, possibly including people on this show, of convalescent plasma in the case of COVID-19, which is obviously what we're going to get to with these papers of yours.
So the answer is relatively straightforward. It is effective, quite effective if you use it early. It's the specific agent is antibody, for God's sake. It differs from monoclonals in that it's polyclonal. It means it has thousands of epitopes, thousands of different types of antibodies. But it is effective if you use it early. The controversy arose because it was tested in conditions when it could not work. COVID is a disease that kills you by inflammation. It was tested on patients who were very sick, and there is no mechanism plausible by which antibody is going to reverse inflammation. It's an antiviral.
So was that done? Our understanding of this disease has evolved. And so were those experiments done at a time when it was not really understood that was too late to be effective?
Well, the answer is yes and no. Early on, think about it, there was nothing available, and people wanted to try it. And they began, and the easiest way to do a randomized controlled trial is to do it in inpatients. Why? Because you can write an order to a unit, to their bedside. What's the problem is that the majority of individuals, by the time they got that therapy, were already, why are they hospitalized? They're hospitalized because they're short of breath. Why are they short of breath? Because they have pulmonary inflammation. So unless you gave it very early, that is at the time of admission, early on, it was very hard to show efficacy. But here at Hopkins, back 3 years ago, we knew that the effective way to do this was in outpatients. And David Sullivan, who's my colleague here, organized this trial. And it required a tremendous amount of effort. How do you give it? They set up tents in parking lots because you can't bring them back into the hospital. They're infected. You're not going to bring them into a suite. These are outpatients. And a year and a half later, he showed that, in fact, if you give it early, it's the expected result, that it reduces hospitalization. It works just as well as monoclonals. It works just as well as the antivirals. And this is now being confirmed by a variety of other trials, including one that was published this week in Clinical Infectious Disease. When they put all the trials together, it works.
Now, one of the objections people sometimes raise to convalescent plasma is the consistency of the product. And I assume that when you're talking about this and this trial at Hopkins, and I know from the papers, you went to some length to make sure that it wasn't just, oh, have you gotten over COVID? Let's take some plasma. It was there was some stringency in testing that this was fairly consistent from unit to unit.
That is correct. A lot of effort was made to collect high titer units. At the beginning of the pandemic, there were no antibody tests. Remember that. So people began just saying, hey, if you got diagnosed, they donated. Now we know that a lot of those units were suboptimal. But for these trials, that's precisely what was done. Now, Alan, you bring up the point of people saying, oh, it's heterogeneous. You know what? Getting a transfusion of red cells is heterogeneous. There are no two donors who give you the same unit of red cells. You can standardize it by antibody titer, and you can standardize it by neutralization titer.
I was going to ask to clarify a little bit exactly what you mean by titer. What is the standard? Is it neutralization titer? Is it an ELISA titer? What is it?
So neutralization titer, as you know, is harder because you either have to use a pseudovirus assay or you got to use live virus. But right now it's automated. But by titer, the word titer means, think about it as a mount. You titer it out. It comes from the old terminology that you dilute it out on a 96-well plate or something. But the way it's done, it gets diluted out. And there is a signal, and the different machines and different tests are all normalized to a different setting. But that technology has been solved. I mean, today you can qualify a unit as having high titer, low titer, don't use it.
And are you using ELISA or neutralization or both?
It's being used. It's machines. Orthodiagnostics uses a solid phase assay. Yeah.
So Arturo, do you have some email program running? Can you turn it off? Because I hear.
Yep, I will.
Sounds.
Here we go. It's unfortunately gonna make noise until it gets turned off, but I will turn them all off. And the only thing that may come is an ambulance. That's the next thing.
That's fine. We can't stop that.
And so, as we're probably going to discuss today, it seems to me that, in fact, the heterogeneity, in particular heterogeneity over time, turns out to be a bonus, really.
That is correct. That is correct. Convalescent plasma is the only antibody therapy that keeps up with the variants.
Right.
And here, Rich is the most, part of me is in immunology, part of me is, me, I'm all over the place. But to me, the fascinating aspect of is what is happening as a natural experiment. When you vaccinate a person with an mRNA vaccine and then they get COVID, they mount enormous antibody responses. And those antibody responses can neutralize variants that don't exist yet. For example, if you had somebody who was vaccinated, who then had the Delta, back in the Delta wave, that plasma contains antibodies that neutralize Omicron. How is that possible? It's possible by the diversification of the immune response. But we have never, in the history of immunology, have an experiment where a host got mRNA vaccines and on top of that got infected.
Right, and this is something that, we know from immunology that this should be happening, but with convalescent plasma, you can actually demonstrate that it's happening in a real viral infection where you get a narrower exposure and then you get a different exposure and then you've broadened the response immensely.
To viruses that don't even exist yet.
Yes.
Which is remarkable. Stop for a minute and think about that.
Arturo,
viruses. Go ahead, Vincent.
The original EUA, first of all, CCP got an EUA originally, right?
Yeah, August 23rd, 2020.
That's
all right. And
COVID-19 convalescent plasma got an emergency use authorization.
Right.
Yes. So, see, it was convalescent plasma only. We didn't have vaccines at the time. Is the EUA been broadened now to include vaccinated people?
Absolutely, no. Absolutely. Because people is defined by titer, and the titers the amounts are unbelievable. That is, what do I mean by unbelievable? It means that the clinical people are saying that you even if you dilute it out, you're still maxing out the machine. We're talking about very high antibody titers, and I think that from an immunological point of view, this is actually fascinating and needs to be studied.
And how long after somebody's been infected is their plasma at these kinds of clinically useful titers?
Well, it varies, but it does drop with time. There's no question that everybody's titers drop, but certainly you have it for many months. And the thing about it is that you can freeze plasma.
Right.
You can freeze it and you can save it for a year, but it is the only thing that we have that keeps up with the plasma, with the variants. And today it's the only antibody-based therapy available.
Is the supply likely to remain adequate for ongoing use? I mean, a lot of people early in the pandemic, oh, I got over COVID, I survived, I want to help other people. And if this was a way to do it, then they would potentially go do it. Are you just plugging into the blood bank and taking plasma from there?
Well, there are many, exactly. The many of the regularly donated units, about a third of the regularly donated units qualify as very high titer. So today, February 25th, the country is awash on high quality plasma.
So
which costs only a couple of $100, by the way. I mean, it's
so as we've actually, first I have a couple of technical questions. You talked about that it's just the liquid after you spin out the cells. Is there anything done to that to enrich for the antibodies, or
No.
is it just the plasma?
No, it's
Just the plasma.
And by the way, it has other stuff in it, okay? It's got endosomes, it's got cytokines. One of the fascinating things about the plasma that is now being documented by multiple studies is that when administered, even to late disease, that it has anti-inflammatory properties. And that may, in addition to its viral neutralization properties. So I think that probably, these are individuals who are resolving inflammation. So there may be other components in plasma that also mediate activity. But I don't want to confuse the audience. It's an antiviral. It's full of neutralizing antibody. And what you're really doing is you're transferring antibody.
And it's important to give that early.
Exactly.
Even it may have some benefit later in disease, but really the key
Exactly. Let's just, right. Now today, Alan, the situation is very different than 3 years ago. Most of the people have antibody. So it's not summer of 2020 when people coming into the hospital, many of them didn't have antibody. So for the immunocompetent, we don't need this stuff anymore.
So
but it's immunosuppressed where the problems are. Yeah, go ahead.
As we've been talking about, there's you can imagine that depending on the history of the donor, there can be a difference in the quality of the product. That is, a person who has just had COVID and not been vaccinated may differ from a person who has just been vaccinated. A person that's had 2 doses may differ from a person who's had 3 doses. A person who's had 2 doses and COVID will differ yet again. In the process of the manufacture, can you or do you try and differentiate those different qualities? And also, the same sort of thing, are the lots tested by individual? Are they pooled? How's that work?
So, Rich, they're always tested as individuals.
Okay.
Every unit and they're tested, today they're tested for antibody content, not for neutralization, although there's a beautiful, if you plot the two, they basically parallel each other. But you're absolutely right. Every unit is different. Every unit reflects the immunological history of the individual. But
so if you want to be sure that you're giving something that covers whatever variant is circulating, okay, is there something in the screening process that provides that assurance?
No, the data are that if you get recent plasma, it covers. The titers are so great. And by the way, this is happening in 2 ways. One of them is the low mass action. So you have so much antibody that even low affinity antibody will now bind, simply because it's just been the old Le Chatelier's principle from first year chemistry. It just drives it forward, the reaction. And the second thing is you have diversification of epitope. So then you tend to neutralize that. But no, Rich is being qualified by the amount of antibody.
And so the product that is used for therapy, is that product provided as plasma from individual donors, or is it pooled, or what? Individual
It's for
donors.
individual donors. And what many physicians do to increase the likelihood, or let's just say to reduce the likelihood of missing a variant, is to simply give 2 units from different people.
Okay.
You're giving a tremendous amount of biological variety when you do that.
And obviously make sure that you're looking at recent product.
Exactly.
Okay. To
Exactly.
maximize covering the variant. Okay. So, how is this produced? And if I'm a physician, how do I get it?
Okay, how it's produced? It's produced from donors. How does a physician? They just have to write an order.
So this would come from a blood bank, basically.
Exactly.
Right?
So the way it would happen is a physician will write an order. The order will go to the blood bank. The blood bank will consult with their network and say, we need a unit of plasma. And then it will be found, it will deliver and given to the patient.
And this is all standard procedure in medicine way pre-pandemic. You can transfuse somebody with whole blood, you can transfuse somebody with plasma, and the blood banks already maintain this type of supply chain for that whole process, right?
That's correct, Alan. Just like we have platelets, we have regular plasma, we have whole blood, absolutely. And you know, transfusion medicine is the most regulated aspect of medicine. And there is a lot of safety that goes into testing these products. The one thing we know is no antibody-dependent enhancement, no cytokine storms, none of that. This stuff is safe.
So nobody's gotten a problem from getting CCP.
Well, let me put it to you this way: you can always get a transfusion reaction, but it's because that happens with regular plasma. There are always blood antigens in there, blood group antigens. But you know, the likelihood of it happening is low, and doctors know how to manage it. Most of them are managed by just stopping the infusion or just slowing it.
Okay. So I presume in the beginning of the pandemic, this was after its EUA, this was used a lot, and then its use declined as vaccines and antivirals came on. Is that correct?
Exactly.
So at the peak, how many people got this in the U.S. at the peak? Do you have any idea?
Yes, we have that data, and it's been published. I can send you that paper. If you're interested, it's in eLife. Put my name on it and put convalescent plasma when it comes up. About 500,000 people were treated.
Okay.
And in the fall of 2020, 40% of the patients being admitted to hospital were being treated with convalescent plasma. I mean, we're talking about industrial use. We went from zero. This country mobilized to deliver that. And that is one of the good stories that you say can come out of this calamity.
Do you have any idea of how many lives were saved by that?
Yes, we estimate 100,000 in the first year. In other words, at the United States, at the FDA, which I give the credit for, because the FDA went out on a limb to make this available based on historical experience. Had they say, no, we can't do it because you got to have randomized controlled trial data, it would not have been used at a time in which we had nothing. 100,000 lives probably in the first year, more deaths.
What about, was it used also in other countries?
Yes, there have been registries for use in other countries, but many countries basically took the tack that no, you got to show it by randomized controlled trials first, and then they set up the randomized controlled trials on very sick patients, and then they say, well, it doesn't work. Look, if you test it in a condition that it doesn't work, and you can't conclude that it doesn't work.
Well, and in fact, one of the papers that you sent us is a randomized control trial.
That's correct.
Where you were the whole team was very careful about structuring the trial so that this was early treatment and used at the correct
early
time.
treatment, double-blinded, control group got regular plasma.
Yeah, and that was from early in the pandemic, right?
Well, David Sullivan got it going here at Hopkins. There was a big group. You can see all the authors that are in it. He is the lead investigator. He put the protocol in April of 2020, and then he managed to clear the IRB and to do all these things. None of this had been done before. To also clear the safety that you're going to set up a tent in a parking lot to infuse patients. but he managed to infuse the first one in June of 2020. And they got over 1,000 patients, and it's the expected result, guys.
So I have
Everything we know.
one small lingering technical question.
Yeah.
Is the blood supply routinely, universally screened for SARS-CoV-2 antibodies, or is there just some fraction that is screened sufficient to maintain the convalescent serum that you need?
The latter, Rich.
Okay.
Yeah, the latter.
I would imagine, because it's expensive.
Exactly. It is expensive, and you want it sort of narrow if possible. You want some people who basically had it relatively recently, so you can be treating the recent variant.
So let me understand. So you don't know if the plasma comes from someone. You do know it comes from a person who's had COVID, right? That's all you know?
That's correct.
Or
So that's now on the donor's questionnaire.
Yeah. That's
okay.
correct.
Okay. And then you said you don't do neutralization. You just do total antibody levels, right?
And that correlates very high. When you get to very high titers,
Yeah.
the correlation is beautiful. The problem always arises at the bottom, where things get very, very noisy.
So let me ask you a question. If this applies to me, so I got 3 EV vaccine doses, right? And then in November of last year, I got COVID.
Stick out your arm, dude.
Wait. This is
Sorry.
the question. Within an hour of my symptoms, I started Paxlovid, and the next day my symptoms were gone. So do you want my plasma?
I think your plasma will be fine. You probably have a very, very high titers of antibody. Because
Even though I took antiviral?
Yeah, you suppress the infection. But the bottom line is that, by the way, you probably would have gotten better. We know with just the immunity you had. What really matters is to have antibody. If you have antibody, T cells are important. Everything is important, okay? But the one thing that is being correlated, I don't want to get into trouble with the audience here. But the one thing that correlates is if you have antibody, you're so much less likely to die.
Right, but if you don't, you can also live.
That's right. Some people do recover in the absence of it.
And, we just looked at a trial, at an experimental study where they gave non-human primates, they vaccinated them, and then I can't even remember the experiment, but basically there was no neutralization, yet they survived because of FC-mediated functions.
That's right.
Right?
And that's not tested.
In fact, one of your papers, you say we don't know the extent of contribution of FC functions because it's very hard to test, right? And in fact, you can't do massive scale-ups to test in clinical laboratories, right?
I always said to my fellow virologists, my fellow friends virologists, I'm not a virologist, the viral neutralization test is the best thing that happened to virology and the worst thing that happened to virology. The best thing, because you can get quantitative data. You can count the plaques, beautiful data. The worst thing, because you completely ignored the other part of the antibody. You ignore ADCC, complement activation, phagocytosis. All those things are going on.
And you ignore T cells as well because they're hard to measure, right?
That's right.
So I keep coming up with technical minutia. We have we've already discussed that the history of the patient is important. Is that actually taken into account in deciding what plasma you're going to distribute, or is it just the titer?
It's a titer, and that's what you want. You want something simple that.
Yeah.
Right. The history of the patient matters only to the extent that you are picking plasma from people who have had COVID within the past 6 months or whatever the question is. But then you're not like detailing what was the course of your disease, did you get Paxlovid, all that sort of thing.
So, does it actually have to be convalescent serum, or can it be post-vaccine serum?
Well, it's the EUA. Basically, the FDA has always basically gone with wanting the person to have had COVID.
Okay.
It's a different response.
And, in order to make that judgment, you have to just rely on their own telling of what their experience is, right?
Right.
You don't actually.
Some people have evidence, but a lot of people still, in relatively recently, went for testing,
Right.
and got a PCR test.
Now that's going to decline. I mean, that's already
correct.
declining, and people are not going to know or maybe even care if they had COVID recently. So are you going to have to alter the process or
So
screen
the answer
randomly?
is probably, Alan, and things are moving, but there is one way to do it, which is you could measure the nucleoprotein antibody.
Now you say in
So
one of your
if you
papers
got the
that
two, you had to have had COVID.
Right.
you say in one of your papers that an issue with that is that, and I wasn't aware of this, that the N antibody response declines over time.
That's correct.
So does that become a problem?
It becomes a problem in the long term, but in terms of population, I think that with COVID becoming endemic, and the fact that we're probably going to be getting shots from now on, I think you will always have high donors.
As a matter
And
of
they're
fact,
needed. They're needed for the immunosuppressed. The immunosuppressed is where plasma now finds a niche.
All right, so that's what I wanted to ask you. So current usage is largely in immunosuppressed patients.
In particular, B cell depleted individuals, they don't have antibodies. So when you give them plasma, you give them something they don't have. And the literature is full of miraculous case reports, people clearing virus after many, many months. See, when you're immunosuppressed, you can't clear the virus, but often you don't make the inflammatory response that kills you. So these individuals can become chronically infected, and they call it smoldering COVID. And it's just you can't clear it. But there have been many case reports of people clearing it.
Have people with B cell deficiencies historically had other infections that were like that would smolder and that could be cured with convalescent plasma?
Well, not convalescent plasma, because that we stopped using it many, many years ago. By the way, it was abandoned it made one contribution to virology, big one. You know what it is?
Jordi Casals?
Yeah, that's how they discovered serum sickness. Not serum sickness,
No.
post serum hepatitis.
I was going to say it saved Jordi Casals' life. Do you know that story?
That's the individual at Yale, right?
Yeah.
Yeah, he got Lassa. He was working with Lassa in the lab, and had just been brought from Nigeria, and he got sick. They brought him to Columbia Presbyterian, and they had happened to have a nurse from Nigeria who had recovered there. They had her plasma, so they gave it to him. And no EUA, nothing. And he lived, but who knows if that saved his life, right? Anyway.
Well, there is evidence, from Ebola and Lassa fever that it's antibody therapy. It makes sense that it could have helped. But
Go ahead, finish.
Well, in the pre-antibiotic era, if you could grow the organism, like pneumococcus, you could immunize a horse, and then you had horse serum to treat it. But for viruses, there was no ability to do that. So they relied on people who recovered. So convalescent serum in those days, because they couldn't make plasma, they used to collect the blood and spin it down. Whereas today, plasma is done through a diffusion membrane. Was used for prevention of measles, for treating even polio outbreaks. People used to take it from their parents and give it to their children. And that, when they began to notice that occasionally when they did this, the recipients got jaundice. And that the tip-off hepatitis virus.
Right. Okay, so then that's where they found the hepatitis viruses okay. Harry Greenberg said he worked on a study where he went to follow up with patients who got plasma or serum or transfusion, whatever, and he said to see if they were getting jaundiced, and he would knock on their doors. He said he borrowed his boss's Mustang, and he drove around the New York area, and he would knock on the door, and he said as soon as the people opened the door, you could see their eyes if they were white or yellow, and you could tell right away. You know Harry Greenberg? Probably not. He's a
Yes, I know him
virologist.
well. We served together, and when I was in the Board of Scientific Council, that's the intramural council. Harry and I served together. I like him very much. Hi, Harry, if you're listening.
So what was I going to say now? Yeah. The thing I was thinking, Alan and Arturo, there are certain people, B cell deficient people. You give them polio vaccine and they shed it forever.
Right.
The oral polio vaccine, and you would like them not to do that. So I'm not sure why we haven't treated them with convalescent serum, right?
Vincent, what has happened in the last 3 years is the greatest deployment of antibody therapies for an infectious disease. I am very hopeful that this carries over to cases like that because it kind of makes sense.
So is this currently underutilized?
Yes, Rich.
Why?
Why education? I mean, think about it. About a month and a half ago, you guys didn't think it worked.
Is that what our error was? We said it didn't work?
I don't know. I'm sure Neil will listen to hi Neil will listen to the program because he always listens, and I think he caught it. That I think some comment was made that that was tried, but there was disappointment. I think you guys did all right. It was just the way that it was.
Well, if
we may very well, I don't know who said that, but I could very well have said something like that because until I saw these papers, in fact, I was under the impression that it just hadn't really panned out. But a lot of doctors
are under that impression. And when you say, what do I spend a lot of my time? Well, we continue to do education. The plasma has no profit, no patent, no pharma. No detail people. There is nobody promoting this stuff. And in a way, you gotta work with your colleagues and try to educate them. And this is why I'm so glad you invited me here, because somebody's gonna listen and it's gonna go to their person and say, did you hear this?
Yes, for sure.
Check it out, please.
All right, so this is what Neil wrote. This was back in November. He said, we didn't talk in any detail about convalescent plasma. We didn't talk about the studies, guidelines for management. And he said, you, Arturo, is one of the chief proponents of the therapeutic potential of convalescent plasma for COVID. He has extensively studied, in one sense or another, therapy for COVID and other infectious diseases. He has repeatedly emphasized that this treatment modality is most likely to be effective for patients with SARS-CoV-2 when used early in the course of infection and when the antibody titer is above an empirically established threshold for neutralizing activity, which does not imply that non-neutralizing activities cannot contribute. Unfortunately, many studies, including some RCTs, did not adhere closely to these not always easily followed guidelines. Okay.
Yeah, so that's the problem.
He wrote it there beautifully.
Yeah. And that's the problem, and I think that's where the confusion has arisen, right. Even with us, is that the trials have to be done right.
Yeah.
Okay.
Right. But if you had tested monoclonal antibodies that work, you would have concluded that monoclonals don't work.
Well, and in fact, I think we even had some discussions about that,
right.
where people were giving monoclonals late in infection and were giving antivirals late in infection, and none of that's going to be helpful in that case.
Yeah,
in my mind,
just
at least,
like Paxlovid doesn't work late
right.
in, right? I mean, it's an antiviral. Please.
In my mind, another thing that happened too is that the convalescent plasma concept was overshadowed, at least for a period of time, by the monoclonals.
I agree.
And they became sort of the first line antibody therapy. But now there's a problem, right? Because with the variants, they aren't working anymore.
Well, we lost them all currently.
So you would like that some patients who are not immunocompromised be treated with convalescent plasmid, right? Because plasma, like I said, plasmid, plasma.
Plasmid.
For example, many patients cannot take Paxlovid because of interactions with the ritonavir, right?
Exactly.
And
But the real problem with the immunosuppressed, the majority of people that have
yeah,
antibody, I am not sure whether giving them a unit of plasma is going to make a difference, right? Especially, but the majority of people with antibody are not ending up in the hospital.
sure. Okay.
Right.
And so you there's a paper which we'll put in the show notes, which is Nature. What is it? Nature Communications.
Nature Communications.
The analysis of anti-SARS-CoV-2 Omicron neutralizing antibody titers in different vaccinated and unvaccinated convalescent plasma sources. And you've already mentioned this finding, but people who have been vaccinated and infected, their sera neutralizes all of the Omicron subvariants as well.
That's correct.
Right? So
Even the Delta one does it. The one from the Delta.
So if Delta infected people who had been
Exactly.
vaccinated, it will neutralize as well. Yeah.
Right. So as a matter of procedure, you want to have recent plasma because it doesn't keep forever. But
Exactly.
in fact, if you have some that's been in the freezer since Delta and Omicron, or if we ever get Pi is circulating, is probably still going to be...
Probably, although that's not going to happen because the Delta was more than a year ago.
Right.
And generally, the goal right now is to use fresh stuff.
Sure.
So this phenomenon of boosting having the effect of covering variants of all sorts that aren't even in the boost, is that because you're boosting conserved epitopes? Reaction to conserved epitopes?
It probably has multiple explanations. One thing it could be, remember the old epitope spreading phenomenon in which you began to recognize things away from the main epitope. So that would be one. Second is the diversification of the immune response. You basically have somatic mutation. You're generating new types of antibodies. Some of those are going to bind. And the other thing, Rich, is mass action. If you have a lot of antibody, even low affinity antibody is going to bind.
Okay.
Flood the
Once
zone.
it binds,
Yeah.
bingo.
Okay. It's fascinating. Just fascinating.
It is, isn't it? I mean, I think that it's remarkable.
This could... So I'm thinking of places where this could be used in addition to immunocompromised patients. Would it also be useful in places where Paxlovid or what have you is hard to get?
That's correct. I mean, where it's hard to get, people are high risk, but I think the majority of humanity now probably has some antibody from the COVID waves and the many vaccines that have been used. So in 2020, this would have been amazing if the world had known they could have easily deployed this stuff now with what we know, and that would have made a tremendous contribution to the pandemic. Certainly, we learn a lot. If we get hit with H5N1, we know what to do, guys. Get high titer and treat early.
Now, one of the barriers to wider... I mean, obviously, people not thinking of this was a major barrier early in the pandemic. But in addition, you mentioned the necessity to test these plasma lots and make sure that you've got the consistency. So a big challenge early in the pandemic was just the testing capacity.
That's correct.
Right? And is that something... How do we address that in a way that's sustainable against viruses yet to be discovered? I mean, we just need ongoing testing capacity.
We need to have the capacity for rapid antibody testing. I mean, in the first 3 months of the pandemic in the United States, many people received units that were not tested. It wasn't until the FDA put out the EUA and said, you got to do this titer, that generally people were saying, well, you recover from it. But I think we got to think forward here. And you got to think about the major classes of viruses that pose threats to humanity. And it's probably a good idea to invest on having some degree of antigen assays that could be deployed very rapidly. so that you could then do this measurements.
So let's go back to H5N1. You said that's what we have to do. But in the beginning of the pandemic, the H5N1 pandemic, there are not going to be many people who have serum.
That's
What do
right.
we do? You have to wait for them to get infected, right?
So sadly, the ones who go first can be treated.
Okay.
But those who recover can help those who follow. And one person can help 2 to 3 people per sitting. So the math is very favorable with a therapy that costs $300.
Okay.
And that's available everywhere,
Everywhere
without
there's
shipping
transfusion
charges
services.
or logistics issues.
right.
If the virus is there, you're going to have convalescent plasma
Right. That's correct.
within days.
All right.
That's
So
correct.
it's available. I mean, we get letters from people who say, I can't get Paxlovid, I can't get remdesivir, I don't want molnupiravir. So plasma can be found in any hospital in the U.S.?
Oh, absolutely. It's under EUA. A doctor, any doctor can write for it in a hospital or in a
Okay.
setting that there is transfusion. And the order goes to the blood bank. The blood bank gets activated and will get you a unit.
All right. Hear that, folks listening. Anybody listening, physicians, patients,
Right.
tell
Well,
tell
and
your
that's
doctor.
not just
Please
the U.S.
educate
that's-
yourself. Please educate yourself. There is no people going around doing education. And this is why this program is so important.
Yeah, we try, right? But, you know, Arturo, part of the problem is that people who spread misinformation have a bigger megaphone than we do.
No. I
Well,
know.
and this is not a misinformation. It's just an uninformation
Right.
issue. That there's not, as you pointed out, Arturo, there isn't a vested billion-dollar interest in promoting this, so it doesn't get promoted.
That's right, and that's right. I mean,
Okay.
but it's got committed people like myself, and it's got allies like the 3 of you, and it's got listeners who can basically hound their doctor. And if you got an immunocompromised patient in your family, please tell them about this, because they need to be able to go to their doctor if they get sick and they said, I heard this. And when the person says, I heard it doesn't work, you say, no, that's not the data.
And the data are, in fact, going to be linked in the show notes with peer-reviewed publications of randomized controlled trials from places like Hopkins. Yes.
Right. And it's recommended by the IDSA and it's under FDA EUA, period.
Yeah.
Okay. So have we fixed our errors, Arturo?
You guys make no errors.
And we do.
Whatever it was, I'm glad that comment led to today.
Yeah, no, we're always
Yeah,
happy
this
to
is
do
great.
that.
Any excuse to have you on is a good one.
I confess to have taken a little side trip here to the and we can put this in the show notes or something, to the Nobel Prize lecture by Emil von Behring for his Nobel Prize. It was delivered in 1901.
Wow.
And it's for therapy against diphtheria, basically convalescent serum therapy that provides an antibody, which is a diphtheria antitoxin, if I got that right. He and Kitsato Shibsaburo were actually credited with really doing some of the initial research on antibodies
Absolutely.
in 1890.
It was done, their original paper was in 1891.
Okay.
And the Nobel Prize, always controversial, they only gave it to him, while they should have given it also to his Japanese collaborator. But I will tell you, think about it, diphtheria was one of the major killers of children. The death is horrible. The parents see the child asphyxiate before their eyes, because a membrane forms in the back. And then all of a sudden, these people came up with the idea that if you could just transfer serum from an immunized host, you can cure this stuff. This was the absolute cutting edge of medicine at the turn of the 20th century.
So one of the real ironies to me now in this conversation is that with all of the high-end technology that we have now, it turns out that an effective therapy that's particularly effective in some cases is, 100 years old, over 100 years old. Interesting.
Absolutely.
Great stuff.
Arturo, can we chat briefly about some of the other things you're involved with?
Yes.
I have this Journal of Virology paper just published. Oversight of pathogen research must be carefully calibrated and clearly defined, with you, Anise Lowen, Jim Alwine, Lynn Enquist, Felicia Goodrum, Mike Imperiali, and Seema Lakdawala. So this is with respect to the recent meeting of the NSABB, where they were asked to recommend some changes. And so you basically responded to each of the recommendations.
Right. That was
correct?
so. We've been in email contact every day. I can tell you that today, Felicia and Mike, I think it was Mike who submitted it, is trying to get op-eds published to basically counter a lot of what's going on. But I remember that when these recommendations came, I said, why don't we deal with them just like we deal with a paper review? Cut and paste them, and let's try to answer one at a time. And you can separate them, and there are some that we don't have a problem with. There are some that we have significant problems with. But at least you can separate them, and at least the criticisms are there, and they are available to reporters. They're available to anybody who wants to be educated.
So let's go through a couple of these. So recommendation 1, amend policy to clarify that review is required for research reasonably anticipated to enhance transmissibility and or virulence of any pathogen, not just pandemic potential, and such that the resulting pathogen is reasonably anticipated to follow the characteristics, to exhibit the characteristics that meet the definition of a PPP. Okay, I assume that
what
one
is
you have a
reasonably
problem.
anticipated mean?
Yeah.
1 in a million, 10%, 90%. It's a flip of a coin.
This is the whole problem, it seems
exactly.
to me.
The language is vague, and this is going to cause, to me, my biggest concern is, look, what is the thing that has worked really well in the past 3 years? Science. Mm-hmm. 3 years ago, we had no vaccines, no antibodies, no drugs, no rapid tests. Science, virology, this is the line of defense of humanity against assaults from nature. Society needs to be working to strengthen it. And if you basically going to put out regulations in which things are vague or something like this, the amount of confusion is going to have a tremendous deleterious effect on our ability to respond to existential threats.
It also occurs to me that this could be deployed maliciously by people who oppose certain types of science, who could claim, well, reasonably, this could be in this category, and you'll always be able to make that argument, right?
Look, everyone in this call, and all of us, we're all for doing stuff safely. What I would argue is that that virology, and particularly dangerous virology, is already under a tremendous amount of regulation. We're talking about a very few experiments. And yet we need this enterprise to be healthy. We need graduate students to go into this. We need universities to want to do this kind of work.
Well, and from the perspective of the people who would be told to review this stuff, so the NSABB is laying out the scope of this recommendation. If you're in a study section and you're reviewing grants, or you're a journal editor, and you've got a manuscript you're looking at, and you've got to decide based on this open-ended standard. I mean, I don't know how you approach that, right?
I think the vagueness of the wording is certainly a huge problem. Reasonably anticipated means something different to each one of us.
Mm-hmm. So one of your recommendations, which I like, is keep oversight at a local level, which right now, if you write a proposal, you have an institutional biosafety committee that looks at it and makes sure there's no experiment there that could be a problem. I think that's very effective.
What I would say is that oversight at a local level has allowed the biomedical research enterprise to thrive and to help us get out of this pandemic. So I would strengthen it rather than think of a new regulatory system. At the local level, people know each other. They know where the problem labs are. They know how things get done.
And there is also, that's not to say that there is no overarching regulation. Those review
Exactly,
boards and such
exactly.
have their own set of regulations that they
That's
follow, which
absolutely
tend to
right.
be quite detailed.
You know, there was an earlier paper that we wrote with the group in which we listed all the regulations that are already in place. This is already highly regulated science.
Well, that brings the question then: why is this being done? You know, it's my impression that there are a few people who make a lot of noise that get these things considered. Do you have any idea why
No.
the NSABB even's looking at this in the first place?
Well, I think, Vincent, that we have just gone through a pandemic. I think that there is a heightened awareness of how, what a respiratory virus can do. Okay, we have gone through that. People are aware of it. People are scared that this is going to happen again. I think that, as you know, there is the continuing argument that the small possibility, because I think everyone believes that SARS-CoV-2 was zoonosis, that this was a lab leak event. And therefore, you put those two together. And I think that's where a lot of the drive is coming from.
It's just the wrong way to be motivated to do something, right? It's like you said, this is a zoonosis, right? So what are we doing? The second recommendation that you had a problem with is remove blanket exclusions from research associated with surveillance and vaccine development or production.
Surveillance, vaccine production, this is society's defenses.
Yes, indeed.
This is how we protect society. Look, we are in the 23rd year of the 21st century. We already had 7 major viral outbreaks. Hopefully, let me get them right. SARS-1, MERS, COVID, H1N1, Zika, Ebola, monkeypox, Mpox, Mpox. 1 every 3 years. What's your line of defense? Science. That's what you need to strengthen. Look what a pandemic with a 1% mortality did to the world. Make it 5%, make it 10%. You know, we can't handle that without a societal collapse. So what we need to do is we need to figure out how to make these arguments and to convince our fellow citizens that the way to protect ourselves and protect our families and protect our future is through maintaining a vibrant scientific enterprise that can respond rapidly. A lot of people here, I went from working on fungi to working on COVID rapidly. We represent essentially the capacity to respond to threats that exist, and in particular to new threats.
And
this needs to be protected. Yeah. This needs to be, we all need to become food soldiers to protect this.
And what they're talking about here, the blanket exclusions for surveillance and vaccine development, this is the umbrella under which many, many labs operate and public health services operate worldwide. And I don't know, when I read this recommendation, I kind of saw it as coming directly out of the lab leak theory nonsense. Because it's, let's clamp down on experiments like the ones that were being done in the Wuhan Institute of Virology, which is exactly what we shouldn't be making more cumbersome, in my opinion.
Look, right now, you cannot exclude the lab leak, but it is improbable compared to everything we know about the biology of these viruses, and given the history of what I just tell you, that the most likely scenario here is that this was an assault from nature.
Well, I would exclude it, Arturo, because there's no evidence for it, and there's plenty of evidence for a zoonosis at the market. So if you keep saying we can't exclude it, people are going to hang on that.
No, because it's the same thing I get asked about, The Last of Us, right? I get, that thing about a fungus turning people into zombies. I get called by reporters and they say, can this happen? And I say, it's implausible, unlikely, but not impossible. I think if you say impossible, then something could have happened, but you're talking about low, very, very low probability events compared.
No, I think the fungus is fine because, but in this case we have no zero. And you listen to Eddie Holmes,
this is something that happens a lot at the interface of science and media and science and law. The problem is if you ask a scientist, is something possible? The answer is always going to be yes.
Right.
I
agree.
It's the nature operates on probabilities.
That's right.
But in practical terms, when you get to the level of probability of a fungus that wipes out humanity the way it's portrayed in The Last of Us, or SARS-CoV-2 coming out of the TWiV, you're at a level of probability where, could you get struck by lightning while buying a winning lottery ticket? Well, it's possible. But
right.
to organize your life or society around probabilities that far out in the extreme is just insane. So we shouldn't.
By the way, fungi make LSD, okay? And they also give you hallucinations.
They do.
Yes.
So is it possible that they will turn you into a zombie? Who knows? No, unlikely.
So in this editorial, there were 11 other points, 11 other recommendations, which you basically had no problem with, right, Arturo?
A lot of them are already instituted.
Yeah.
A lot of them are already instituted. They basically, it's the things that we do. It's a standard practice. They're basically recommending that standard practice be codified. I don't have a problem with that. We do it already.
It's totally appropriate for these rules to be reviewed by a group like the NSABB from time to time. Is this adequately, are the mechanisms for this adequate? Do we need any more? Do we need any less? And so a lot of their work is actually totally appropriate.
So maybe you've already said this. What's the status of these recommendations now, and what's likely to happen going forward?
Well, first of all, they're recommendations. They go to the government. The government will now take them. They can take all of them. They can take none of them. They can add to them. They can change them. And I think what will happen at some point in the future is that they will be published in the National Register, and then we will have a chance to comment at that point. We would like to see what they are, and I would say let's wait and see. In the meantime, let's continue to make the positive case for science, where there is no controversy.
Who is they in the government that's going to review these?
This would be multiple agencies.
Yeah,
this would be, some of these are going to fall under the jurisdiction of FDA. Some of them are going to be CDC. Some of them are going to be the Public Health Service. Some of them may actually be DOD. So it's going to be multiple federal agencies that would presumably look at these recommendations. And the process Arturo just alluded to, there is a very specific process for federal rulemaking. Say the FDA would publish something, or the NIH would publish something in the Federal Register saying this is a notice of proposed rulemaking. We propose to regulate this research in this manner. Then there's a comment period and a period of review, and there's a whole back and forth. There's a lot that has to happen, and there are a lot of opportunities to comment further on this. So I am somewhat reassured that the process will involve a lot of people who are not just the NSABB.
Right. I agree. I agree. But we can continue to make the case for science.
Yes.
Sure. And that is really, really important.
Arturo, anytime you want to make the case for science, let us know, and you can come and make your case here. We have a lot of listeners. One more thing. The other article you were part of, Virology Under the Microscope, a call for rational discourse. And, Felicia Goodrum, the first author, was on a few weeks ago. But tell us your view of what is embodied in this this commentary.
So, I mean, my sense is that we are living at a time in which virology is under attack. And what I have basically said to other scientists is that this is an attack on the greater science. That the tools that there is being attacked are tools to establish causality. You need to be able to establish causality. These are the tools of science. And what we say is that all these things that we've been talking about, they need to be essentially confronted and discussed. And we can do it. We can do it.
This is the article that has your wonderful tables in it of
Yes.
the diseases for which science has provided vaccines and antivirals.
Yeah, there was a
And-
whole discussion as to whether one should count smallpox because of vaccinia. But at the end of the day, virology puts it in a vial.
And the other table is the examples of useful gain-of-function experiments. I really like that. Very nice.
Arturo, this is my view of what happens. Most people are too busy, so they don't talk to reporters when they call them. So the reporters find a pundit who has no idea what they're talking about, but they have an MD or a PhD, and they talk to them, and people think they know what they're talking about, and then bad stuff spreads from that. That's part of the problem.
I agree, but you know what? Well, this is really important. And we need to take time of our day to talk to reporters, to write op-eds, to write letters. Whenever you see anything like that, write a letter. These arguments need to be met.
And when reporters email you, email them back.
Absolutely.
Just putting that plug in there.
I always
it would make my
always
job so
try-
much easier.
I always try to talk to the media. It's our job. After all, the people are paying for this research.
So,
go ahead.
I have a student in my virology course, which I teach every year at Columbia, right? She's been a journalist for 25 years. And she said to me, I'm taking this course because I recognize that virology coverage is abysmal and I want to fix it. So I said, this is great. This is exactly because most of them say, I don't have time to learn virology. And that's part of the problem. I say, read this article. I don't have time to read it. I have a deadline. It's not an excuse. So she's doing the right thing, and everywhere else, please follow.
I really another plug for this article is you have a couple of tables here that describe or figures that describe the regulatory process.
That's right.
Both at the local and the federal level, and also the peer review process. Okay,
Exactly.
That assures that we are as accurate as we can be and as safe as we can be. And those are really nice, concise diagrams of this kind of stuff that the general public is probably oblivious to or don't even think about.
So
They
those
just
articles
assume,
become really important, Rich, because somebody says to you, well, we need to regulate this stuff. It will take you a long time to find the regulations, but now you can pull that article and you could send it to them and say, look at the figure.
right.
The thing that I love most about this article, I mean, it's well written and all that, but the tables and figures and the data that you put into it and consolidate it in one place. This is an incredibly useful reference and
really
will
nice.
remain so for a very long time.
All right.
Thank you.
Arturo, we'll let you go. Arturo Casadevall, Johns Hopkins, Bloomberg School of Public Health. Thank you again for coming on. We appreciate it.
Thank you for having me. I'm going to hit the red phone. And,
Okay.
And don't go till it stops unloading, right? Hitting the red phone. Bye-bye, guys.
Take
Bye-bye.
care.
Well, that was great.
That was great.
So convalescent plasma is good.
Yeah.
Yeah.
Okay.
It's got to be the right stuff, and it's good when used properly.
When used properly. Yeah.
Yeah, and you
Gotta
have to
give it at the right time.
Yeah, you have to use it properly. That's right. Okay, let's do some email. Alan, can you take the first one?
Sure. Charles writes, hello all. 65 Fahrenheit, 18 C, and damp. Not a great day, but 65 in mid-January is both nice and something to complain about. Not sure if this is a TWiV, Daniel Griffin, or TWIM question. The subject that brought this to mind was most recently on TWIM 279, but has been on TWiV before, and with amoebas being parasites, how could I not ask Dr. Griffin? That subject is successful phage therapy. I remember summer stories about Naegleria fowleri amoebae eating people's brains, and there not being much that would help them. Antibiotics may help early, but nothing seemed to work late. So thinking way out of the box, I remembered that giant viruses infect amoebas. So is that like phages infecting bacteria? If so, could we find or modify, please don't let Rand Paul know, a giant virus that inactivates Naegleria fowleria amoebae? Is this interesting, or should this computer programmer get back to his lane? Thanks, Charles. P.S. Even if you suggest I get back in my lane, history suggests I will not follow your excellent advice. All right, Charles. Well, you don't have out of our lane is comfortable for us. And I think this is a fascinating idea. I mean, I can see obstacles to it. The amoebae are in the brain. Getting anything into the brain across the blood-brain barrier is a massive challenge, even if it's like a small drug. There's a lot of stuff that just doesn't cross that. So getting a large virus through, I don't know, but gosh, neat idea.
Maybe we could make the virus smaller.
Yeah?
No, it's a good idea. But yeah, I think you hit on the main problem, that getting the giant virus across the blood-brain barrier is going to be a challenge, right?
Yeah.
Yeah.
So to answer your direct question, yes, this is like phages infecting bacteria. Giant viruses infecting amoeba is like phages infecting bacteria.
Well, and is also like animal viruses infecting animal cells.
Yeah.
Amoebae are not bacteria. They have organelles. They are eukaryotic cells. So it's really a little more like pox virus infecting a human cell.
So with the exception that you're talking about a unicellular organism, the amoeba.
Yes.
Okay.
Mm-hmm.
What comes to my mind is that I'm not sure what the host range of many of the giant viruses are, whether they'll affect this particular amoeba. But, that doesn't mean you couldn't go screening for giant viruses that do infect this amoeba, and I'll bet you'd find them.
And I would say go find Naegleria, and I don't know how culturable it is or anything about who's working on it, but look for giant viruses in it, and barring that, in its environment.
Problem is this is a rather rare affliction, right?
It's extremely rare, yes.
Not sure that the pharmaceutical companies are going to get in line to develop a virus for this. That's the problem.
But the basic research on it, looking for giant viruses that infect this amoeba,
is
a cool idea.
The giant viruses are largely found using Acanthamoeba castellani, I think. Which is a amoeba you can grow in the laboratory readily. So,
right.
But it would be interesting, yeah. All right, Rich.
I'm looking at this amoeba in Wikipedia, and I'm scanning down. I run across a picture of the Roman baths in Bath in the UK.
Yeah,
because that's one of the places that you wind up getting this thing, drinking water that's got it in it. Anyway, excuse me. Flavia writes, after listening to many, many TWiVs over the past years, I, a junior college graduate only, whose interest was literature and beginning to grasp virology. Your podcasts are fascinating and thankfully necessarily often repeat concepts and accompanying vocabulary, and that repetition seems to have pierced my veil of ignorance re virus matters. Thank you. I write today because your most recent TWiV included mention of splashback toilet flushing problems, even potentially with urinals. I had just come across the linked article and thought I'd pass it along for your possible, albeit unlikely, interest. Gives a link, says, I wish TWiM were both audio and visual. And let's see, I haven't looked at this link.
Very funny. This is great. They designed urinals that don't splash as much.
Oh, cool.
Because I was talking about how you couldn't put a lid on a urinal like you could on a toilet when you flush it, right? So they looked at how dogs pee, and they figured out the angle of the urinal to prevent splashback. This is
I
just
love the
great.
quote, the
So wait a minute,
idea
are dogs
originated
evolved?
exactly where you think it did.
What was that? Sorry, Alan. What?
The idea originated exactly where you think it did.
So are dogs evolved to have a minimal splash pee somehow?
I guess.
God, this
'Cause,
is
you
even
know,
got a video.
It's a problem with pants. You get splashback on your pants for sure. Interesting. Anyway, this is great. Thank you so much.
Yes.
I'm looking at the video.
Huh.
I got drops splashing.
I like it.
My goodness. Oh, here's a way to... The little things in the urinals, the little spiky things,
Yes.
those reduce splash. I'll be darned.
They do.
Yeah.
Well, also
according to
the
this
problem
video.
is flushing, too.
Yeah.
Right. The original discussion was the aerosolization on flushing.
So this apparently it cuts that back too.
Great
you know,
video here.
You can't get away fast enough to avoid the aerosolization if you can't close
Right.
the lid. Interesting. All right. Ginny writes, I just had to share, in case you hadn't seen it, that Fox News wondered if that Chinese balloon 60,000 feet in the air was filled with virus, came from Wuhan, and we'd all end up back on ventilators. I literally laughed out loud. They really don't get science, do they? Hope that gave you a laugh too, Ginny from Rochester, New York. Of course they would say that, right?
Yeah.
That's pretty funny.
Oh. Yeah, funny in a really
In a
depressing kind of way.
Dark and depressing
Yeah.
way, yes.
Yeah. Yeah, it is.
It's all about the xenophobia, folks. Yep.
Yep. It's very sad. I agree. All right,
Yeah.
back to the top.
By the way, on the subject of balloons, did you see the thing about the hobby ballooning group? This came originally across my ham radio email lists because there's a big crossover there. People launch weather balloons quasi-recreationally, right? And they'll put little ham radio transmitters on them so they can track them. And one of the balloons that got shot down by the Air Force after the big Chinese spy balloon, they went around, then boosted the sensitivity on all their sensors and shot down other balloons. And one of them was most likely one that was launched by a hobby group just to do it and to experience, that near space flight path. And it had circled the globe something like 6 times.
Man.
And then apparently got
Wow.
shot down.
Too bad.
Did you see the cost analysis of shooting down one of those balloons?
Oh, yes.
Costs like about $500 million.
Yeah, the
missile
it's like
itself
a
is expensive.
$400,000 missile,
Okay,
the Sidewinder.
$400,000.
Yeah, that was shot at it.
Alan, you're next.
Okay. Hunter writes, esteemed Twivers, unfettered praise seems to assist in getting an email read during the podcast. All of you are beacons of light on coronavirus. Thank you. Well, actually, I'm not sure that the praise is what's getting the letters read.
They
But
all
anyway,
have praise. That's why.
A lot of them have praise.
boss.
Alan said to keep it succinct, so here goes. This link is to the annual report for rabies surveillance in North America for 2020. Give the link. This shows over 87,800 animals tested in the U.S., 11,600 animals tested in Mexico, 2,600 animals tested in Canada, so a total of about 102,000 animals. Twivers, as well as many of your guests, have bemoaned the lack of animal surveillance in general, and as it might relate to establishing a reservoir of SARS-like viruses in North American wildlife. Thrilled to have Angela as part of the group. About time veterinarians are in the mix. Of the 95 people David Quammen bios in his book, 10% were veterinarians. All hail the 10%ers. My questions for TWiV are, 1, how long might the coronavirus remain viable in these specimens? Generally, they are shipped in short order on ice for the rabies screening since people would like to know ASAP. 2, the entire animal would not be available. Okay, so let's do these one at a time. If you freeze it, the virus should be viable or certainly assayable for days, weeks.
Yeah.
I mean, as long as it's kept on ice, right, or dry ice would be ideal.
If it keeps rabies active, it's likely to keep coronavirus active,
Yeah.
I would think.
Yeah, so I don't think that would be a problem. 2, the entire animal would not be available for a sample taken in larger species. Don't send us the whole deer. But in most species, you will get an intact head, not with a bovine or equine. Check this out. For brain removal with compressed air, we use the water hose method. Okay. Does the head provide a suitable area to swab, say nares and sinuses? Yeah, I guess you could do a nasal swab on a head. Right.
Yeah, and that deer study that we used a lymph node from behind the nasal cavity, as I recall.
Yeah.
That they had shown in previous work was a hotspot for SARS-CoV-2.
Right. 3, since SARS-CoV-2 most likely arose in bats, it seems like more bat surveillance, in addition to testing rabies-submitted bats, should be occurring. Is it going on in the U.S. currently? The study shows that for the U.S., over 24,000 bats were submitted in 2020 for rabies testing, so a ready pool might be available to sample.
Yep.
I do not know if U.S. bats are being routinely or even periodically tested for SARS-CoV-2-like viruses. I haven't seen anything on that.
Nope.
I just Googled and I got baseball bat testing.
No.
I should have put in Chiroptera.
No, don't send in those kinds of bats.
Yeah, there's not much surveillance in the U.S. at all. That's what I've heard. I heard that at the bat meeting last summer. Yeah. But these, from this study, he says you could get 24,000 bats. You could test those for SARS-CoV-2. Yeah, unless if they were not kept frozen, well, they'd be frozen or thrown out.
They're being tested for rabies. Yeah.
Yeah.
4, if the virus is stable for a length of time after the animal expires, how about large animal veterinarians swabbing all the roadkill we see every day? I will leave the skunks for the COVID-19 patients whose senses of smell and taste have not returned yet. Opens up the potential to recruit hunters, trappers, and other outdoor enthusiasts in the testing program. Sure. I would think for deer, hunters would be, ideal.
Well, that's the study we did that we're using.
But that's,
hunted deer, but
for other animals found dead in the woods or on the road, I don't know. I guess if they're going to be tested for rabies, I think the way to do this would be to just take everything that's being tested for rabies and piggyback on coronavirus testing, broad spectrum.
So the problem I see is if you tell people to bring in your carcasses, then you're going to have a containment issue, right? Because potentially infectious material is now going to be concentrated, and people are going to potentially expose themselves. I think
I wouldn't be so worried about the receiving lab containment because they're already receiving samples that they need to deal
bringing
with
it in.
containment
I'm worried
on.
about.
But it's the public, You don't want people bringing in dead birds because then they get West Nile virus, etc.
I'm going to back up a little bit on my previous comment because I said anything that you can test rabies on ought to be good for SARS-CoV-2. That I'm thinking they were testing for active rabies virus. I guess I said that. Okay, I don't know how the rabies tests are done. Okay, because if you're just doing PCR or something like that, well, still that's an indication.
Yeah, I think
you
that's
find
fine.
SARS-CoV-2 in something by PCR. That's a good indication.
I think PCR is good. It's fine for this. Yeah.
In fact, we get to that in the next one. 5, are the primers to detect coronaviruses readily available? I'm thinking if the animal tested is negative for rabies, would not want a positive rabies animal around any longer than necessary, then the animal or sample could be tested for other viruses with an emphasis on coronaviruses. You can actually design primers for a wide range of, right? I mean, you just pick conserved sequences.
Yeah, and you could multiplex them. You could do them all at once. Sure. That's
I
no
think
problem.
coronaviruses, flu viruses, a few other families. You could design a multiplex test that would just be done as a viral panel on maybe not every sample, maybe randomly chosen ones or ones that test negative for other known viruses.
I think this would be a great the as you will see in the next paragraph.
Yes. This is all pie in the sky, most likely, since it would require funding and a commitment to long-term One Health pandemic surveillance, which seems to be difficult to maintain.
Yep.
Yes, that is what that comes down to. 34 degrees Fahrenheit, blizzard conditions, and that was just a few weeks ago. Hunter is a veterinarian, in case that wasn't obvious, in Prairie du Sac, Wisconsin.
Just a quick look. It looks to me like, you can probably buy SARS-CoV-2 specific primers from any of a number of biological supply companies.
That's easy. The problem is, Alan, that if you found something, then they'd say you introduced the next pandemic virus by discovering it. You can't win. You cannot win. This is a real problem world that we're living in. And you guys know, of course, that Idaho was going to pass a bill to make it illegal to give mRNA vaccines.
What
is this?
You guys haven't heard this?
No.
The Idaho legislature is considering a bill to make it a crime to give an mRNA vaccine because they haven't been tested properly.
Oh, God.
Wow.
Just Google Idaho mRNA vaccine bill. Idaho lawmakers want to criminalize mRNA vaccines. So they introduced a bill, 154. This is
okay. So two lawmakers introduced a bill.
That's fine. It's still a screwed up situation that two lawmakers think it's a crime.
Yes. No, I don't anticipate this going anywhere.
It's still screwed up.
Periodically, yes, you get some real pieces of work in office.
Yeah, I would say every year you get real pieces of work.
Idaho listeners, if either of these idiots are if you're in their district, just see if you can get rid of them at the next election.
All right, next one, Rich.
James writes, hello. Interesting to come across this, he gives a link, after listening to you and Dr. Griffin on this subject of peer review in light of the infodemic during the past few years. This process needs examination. A faithful listener, James, and he links to a Nature feature that says, titled, Stop the Peer Review Treadmill, I Want to Get Off. Faced with a deluge of papers, journal editors are struggling to find willing peer reviewers. I have not read the article, but I can imagine.
Hey, Alan, do you know anything about this?
Yeah, I've certainly heard about this. So the problem is twofold. Well, more than twofold. There's been this increase in number of submissions and decrease in available time for researchers to review the papers. This became really acute during the pandemic because everybody suddenly became a SARS-CoV-2 virologist, and there were lots and lots and lots of papers being submitted, and the people in coronavirology were overwhelmed with peer review requests. But it has now, I don't know if it's really expanded or if this is just a long-term trend that is coming to a head now, it's hard to get peer reviewers. And the journal proliferation has been another component of this. We have a lot more journals than we did even just a few years ago. So people are being asked to review for lots of different places. And I think that's, yeah.
It occurs to me that many of our listeners may not know how this works.
Right.
So you submit an article to a scientific journal to get published, and an editor, somebody in that journal, looks over the article, and there may be a preliminary screening process to determine whether or not it has any merit at all and deserves a little attention. And if so,
well, or whether it fits that particular journal. I don't want people to think that there's, just because the editor desk rejected it does not necessarily mean your work is without merit. It just means they don't think it belongs in their journal.
And then that editor identifies a number of scientists to send the paper out to, whose charge it is to read the paper and comment on whether or not they think it's valid and significant and et cetera. And although the editor on the journal may get a stipend or may get paid for what they do, the reviewers do not, generally, okay? So this is entirely a volunteer effort. Scientists who are, university scientists or research scientists in any of a number of disciplines sort of assume that this is part of their job, is this pro bono work. There's a bonus in that is you get to, it helps keep you up with the literature, but it takes time, and it takes a lot of time. So you can imagine that in a situation like a pandemic where there's a proliferation of papers, you can get to the point where you're spending all your time doing reviewing, and you got to put a stop to it.
You don't review anymore, do you, Rich?
No. Probably within the last year, I may have reviewed a paper or so, but as time has gone on, first of all, I have less and less inclination to do it.
Yeah.
And I also don't feel as qualified. I've gotten a lot of traffic during the monkeypox outbreak, but a lot of it's not really appropriate for me anyway.
You don't miss it, right?
No.
I don't miss reviewing grants, that's for sure.
Oh, no. The one thing I liked about grants was I actually enjoyed chairing a panel.
Yep.
Because I felt like, first of all, as the chair, I had to pay attention. And that actually, although a lot of people thought that would be, maybe I even thought initially it might be a huge challenge. It wasn't, because otherwise you're trying to pay attention, okay, and it's tedious and it gets boring. But if you know that when this discussion is over, you're going to have to summarize it, that's a different story.
There will be a test afterward. Yes,
that's right.
So I enjoyed that. I also found that I had an opportunity to, I don't want to overdo this, say to influence the process, but at least an opportunity to give my perspective on some of the issues that came up. Like, for example, whether something absolutely had to have a translational bent to it, okay?
Yeah.
So I had a platform to promote basic science, and I appreciated that. It was good. No, I don't review anymore. I don't miss it.
Paul writes, great issue on Sunday. As always, really enjoy spending a little time with you folks. To the point, I understand the concern with unclear statements asserting that certain gain-of-function experiments shouldn't be conducted when there is a reasonable expectation that risky outcomes might occur. There's that word again, a reasonable expectation. Yeah. Yes, this type of unclear constraint could greatly hamper needed research. Such ambiguity is unfortunate. But when I consider specific instances, this kind of statement makes more sense. Additionally, this type of statement invariably proceeds greater restriction because a few parties will undoubtedly exercise questionable judgment. To insert or suggest the insertion of furin cleavage sites in a sarbecovirus that might increase infections in humans is poor judgment. To insert or suggest the insertion of a receptor binding domain into sarbecoviruses with high human ACE2 affinity is in poor judgment. A reasonable informed person would not partake in such experimentation. The risk-reward benefit cannot overcome scientific curiosity in these cases. Virology and other biosciences currently enjoy a degree of freedom and self-regulation that will shift to a more constrained framework, much more restrictive than desired if the average taxpayer perceives a lack of self-awareness to the significant risk in pathogen research, no matter how small the possibility of accidental escape. As you guys mentioned, it's not the protocols that are the problem. It's the human beings make mistakes. You guys are great, Paul. Thank you, Paul. I would just say that to insert and suggest the insertion of a furin cleavage site in a sarbecovirus that might increase infections in humans is poor judgment. We actually didn't know that previously. So people did insert furin cleavage sites in various viruses. with various outcomes, but nobody put them in people, so you're never going to know that. But we had no idea that it would increase infections in humans. We now know that the site appears to increase infectivity in humans. It appears to. However, we wouldn't have known that ahead of time. So it's kind of in retrospect, it's not fair to criticize it because you wouldn't have known that. That could be a dangerous experiment. Anyway, thank you, Paul. Let's do one more round, folks.
Okay.
Alan, please.
Sure. Catherine writes, "Hello, TWiV gang. I was listening along to TWiV 983 when I heard Vincent exclaim that the high price of baguettes was possibly related to the increase in the price of eggs." Oh. Yeah, I wasn't on that episode.
You would have remembered that.
I would have remembered this.
Alan, I bought a baguette last week. It was $12. And so Dixon was here.
Where the hell are you buying a $12 baguette?
That's what everybody asked me. So I said to Dixon, that's because of the eggs, right? And he goes, yeah,
for 99
probably.
cents each.
So Dixon said, yeah, it was the eggs. I didn't know if there was eggs in baguettes.
No, now challah, that would be affected by the eggs.
Yeah, challah's got eggs. You're right.
Yeah. Okay, I haven't read this email yet, but I'm sure that's what we're getting to. As someone who has devoted a great deal of my time to eating French pastries and other baked goods, let me assure you that a real French baguette may not contain anything other than flour, salt, water, and yeast. There are never any eggs in it. It is likely that the war in Ukraine and the subsequent destruction of wheat harvests there was causative. It could also have been linked to the war in Ukraine and the subsequent spike in natural gas prices, which causes shortages of fertilizer made from natural gas. Likely all of these combined together with the increase in the price of housing for the people who were making those baguettes for you. Sincerely, Catherine, who did her thesis in France and ate very well there. Yes.
So that's the thing. Don't ask Dixon about food because he doesn't know what he's talking about. I assumed he would know, but I certainly don't know because I don't really cook. So I don't know why it seemed a lot of money for a baguette, $12, right?
That I think we can all agree on. That's a lot of money for a baguette. I think you got played.
Okay. Thank you. And many people wrote in, so thank you all. This is just representative.
So you got a bunch of baguette mail.
I got a bunch of baguette mail.
Yeah.
Rich.
Jerry writes, "I'd like to shed some light on the car-deer collisions based on my experience at the Cooperative Wildlife Research Laboratory at SIU in Carbondale, Illinois." That's Southern Illinois University. When I was there in the 1980s, one of the master's students did a research project on that topic. Use of night vision scopes to observe road crossing behavior, surveys of drivers involved in deer collisions taken from all accident reports, snail mail era, and incidents of collisions on roads where hunting occurred in fall versus non-hunted areas. Very nice write-up and presentation. This is the stuff that goes into the body of knowledge used to manage deer herds and doesn't get wide publication. The first item of importance was that over 90% of the drivers never saw the deer until it was in front of the car, too late to avoid. Use of warning signs indicating deer collision area had no effect on this. Side note, the signs are up because people demand them, not because they work. On behavior, a lot of collisions involve doe-fawn interactions when crossing a road. With oncoming traffic at a distance, the doe will cross, but the fawn will hang back. When the car gets much closer and will clearly come between the fawn and the fawn makes a dash across the road. Most times he makes it, but not always. They make it, but not always. He spent many long nights with a night scope on local roads watching the process. In fall, at the onset of rut, there was no difference between the hunted and non-hunted road areas in terms of collisions. They both have an equally proportionate increase in accidents. Hunting restrictions aren't going to reduce the numbers. There was pre-DNA gender identification, so we didn't have reliable data on male-female ratios. A lot of times there was just a smear on the road having been run over by a semi-tractor trailers and cars a number of times. Jerry. All right, so that fills us in on roadkill.
Good.
Deer roadkill. Good. Excellent.
Yeah, so,
thank you.
this last bit. So basically, hunting doesn't make a difference in terms of road accidents, right?
Yeah.
That's kind of what I would expect.
All right.
Because the deer, I mean, they run from hunters, but that's not their whole thing. They're, especially during the rut, they've got other stuff on their mind. But all year, I mean, they're moving around their own way, and they will cross the road when they cross the road.
When did this come up?
Or they'll cross the runway. There are a lot of, if you go to the FAA database on animal collisions, people think bird strikes, but you find actually there are a lot of land mammals in that category.
I think it was our last Friday was our deer paper. Deer.
Yeah.
Mm-hmm.
I remember that. I just don't remember this particular discussion, but that's okay. I was probably
we did have a little
somewhere else.
discussion about collisions, I think. Yeah.
Okay.
Maybe you were staring into the headlights. At the time.
Right.
It could be.
All right. I'm going to do two here because the first one is short. The first one is from Jerry, who writes, I don't know if this story itself will be your thing, but be sure to read the story behind the story at the end. All right. So this is a Nature article called Not Too Soon, Fear is Contagious. And it's by Jerry Lean. All right. The author is Jerry Lean, who happens to be the author of the email. And it's about fear during a pandemic, essentially. It's very good. And then you get to the end, the story behind the story is very cool. It says, the last paragraph, many thanks to This Week in Virology podcast gang for keeping me sane throughout the pandemic with clinical updates and actual science on the virus.
Excellent.
So thank you, Jerry, for the mention in the article and for writing in, because I wouldn't have seen it. It's really good. I would read it. It seems to be open access.
Yeah, Nature has started doing these sci-fi short stories.
Really?
Yeah.
That's good.
Yeah.
As long as you realize it's not science.
No, they are very obviously. I've read a couple of them, and they're quite obviously fiction.
What did she write? She's apparently it says so in the article. She wrote this before the pandemic, and then she held on to it. She said, I sold this to Nature in January 2020. It was a different story then, because we were before the times when pandemic was dark science fiction, not everyday life. And then she revised it. It's really good. Really good. Thank you, Jerry. And one more is from Drew. I heard some nostalgia expressed for the old Weather Channel feedback with smooth jazz on the last epitope. And I thought I'd share this gem I came across recently because I also have a soft spot for the old format, and gives a link for that. We'll even play lo-fi smooth jazz on a loop. Thanks for making the show, and I hope this delights you all as much as it does me. Drew from Minneapolis. We're about to get another foot of snow. That's very cool. Thank you. Yeah, last week someone was talking about the Weather Channel used to have this jazz, and apparently you can still play it. Very cool. The things our readers come up with for us.
Great stuff.
Excellent.
Let's do some picks. Rich, what do you have for us?
I have picked a Wikipedia article on the Flammarion engraving. And you got to click on this to know what I'm talking about. This is, I don't know why I had this in my TWiV pick pile, other than you see this engraving from, let me see, it's dated to, I got to look back here.
1888 was the first publication, but there's an open question about who created this engraving.
The Wikipedia article favors the attribution to where it was actually first published, and that is Flammarion himself. Okay, it's Camille Flammarion. He was an astronomer, but also a writer. He wrote a lot of science fiction and stuff, French. Okay. And this is a very familiar, I mean, you've seen it all over the place, a very familiar engraving that has, the picture is of an individual who has apparently crawled to the edge of the earth where the firmament, that is the shell that is over the earth, meets the edge of the earth, and he's sticking his head and his shoulder through the firmament to see what's on the other side. And on the other side are a bunch of clouds and suns and stuff, and an object called, now I'm going to mess this up, it's this wheel that's Ezekiel's wheel. It's there's a special name for it, but it's essentially the wheels on God's chariot. All right. And there's the context in Flammarion's book is quoted in the Wikipedia article, and down at the bottom of it is the important quote. I mean, basically, the idea of the firmament was around for a long time. The notion that, and this is what really interests me, the notion that we live in a bell jar, okay, and there's this sphere, there's this sphere over us called this firmament, and all the stars and stuff are on the firmament, and there must be something behind that. In the biblical sense, it's holding back the waters, and it helps separate the land from the water. So those would be,
and
I suppose,
in more
water.
detailed views of this cosmology, there are multiple spheres to encompass the stars, which follow one pattern, versus the planets, which follow, and the moon and the sun. And so you can accommodate these movements by nested spheres that you're looking at.
But it's interesting to consider that in that particular cosmology, you ought to be able to walk to the edge of the Earth and find where the firmament meets the Earth, and then maybe try
lift it up and look under.
That's where lift it up and look under. So the last phrase of this context is good. A naive missionary. This is from Flammarion's book. A naive missionary of the Middle Ages even tells us that in one of his voyages in search of the terrestrial paradise, he reached the horizon where the earth and the heavens met, and that he discovered a certain point where they were not joined together, and where, by stooping his shoulders, he passed under the roof of the heavens. So I need to get myself a print of this and put it up somewhere. I think
curiosity.
it's great.
Yeah, glimpsing a new world from the one that we're in. It's,
That's what we do as scientists.
Exactly.
Cool.
Maybe that's why I picked it.
Probably.
It's science. This guy's looking through the firmament.
Rich, what do you have? I mean, Alan, what do you have for us?
This is my fourth of what are going to be the series of 5 story-driven video games. And people could comment that there are a lot of wonderful story-driven games I'm not picking. I'm picking the obscure indie titles that people may not have gotten around to playing. This is What Remains of Edith Finch. Mm-hmm. And I played this on Nintendo Switch, but it's available on everything. I'm linking to the Steam page for PC users. And this is it's kind of an episodic story, but it's told from the perspective of the last surviving member of this family, the Finch family, which supposedly has a curse that various members of the family die in strange ways. And so she goes to the old family house, which has been abandoned ever since the last death. And you are playing from a first-person perspective as Edith Finch, visiting the various rooms in this house, which you can unlock sequentially. And what's amazing about it is the way in which everything is integrated into the stories being told. The gameplay and the stories and at one point you're playing as you enter the rooms, you play as the individual who used to live in that room and then met their untimely end. And in most cases, you're playing a sequence of events that ends with that character dying. But one of them, you're a baby in a bathtub, and the gameplay is moving the toys around in the bathtub. And I'm not going to spoil anything, but it's just integrated into the way the story is told in such an amazing way that could only be done as a video game. And I just think it's really, really cool. Even if you're not into games, if you have any kind of platform that can run this, I highly recommend giving it a try.
Can I just run this off a PC?
You can run this on a PC. You can install, the easiest way to do this would be to install the Steam app on your PC.
I see. There's even a little click up here on the website.
Install the Steam app, and then you can buy. Oh, it's actually on sale right now. I hadn't even timed this. You can buy it for $5. It's 75% off, and then you should be able to play it. It's not super processor intensive, I don't think. So most relatively recent machines should be able to play it. As I say, I played it on the Nintendo Switch, which is a pretty low-powered, computing-powered device.
Cool.
Cool. Neat. Nice. Someone sent in an email last week about a game for you or something.
Oh, okay.
Take a look at it.
Yeah.
All right. So I have a. I'm sorry to pick a Wall Street Journal article because I know it's behind a paywall for most people, but I got to read a good amount of it to you so you know what it is. It's called. It's an opinion piece. The CDC's Long COVID Deception by Alicia Finley. And for you guys, I put a link to a PDF that I have made. So someone sent me the original PDF, and so you can have there it is, your own copy, Rich and Alan. So basically, this is about a study done at George Washington University, published in Emerging Infectious Diseases. They claim that a recent study of students on campus showed a 36% rate of long COVID in the students. And the study concludes that young, healthy people face a high risk of chronic debilitating symptoms after infection, despite being at low risk of getting severely ill by the initial infection. And I picked this because the writer has got it. She got it, that this is a bad study in many ways, and I'm very impressed that she got it. She says the study also finds that the unvaccinated were more than twice as high a risk of developing long COVID as vaccinated. This sounds plausible, but drilled down, it becomes clear that the evidence is too thin to draw any conclusions. So basically, GWU held classes during the first year, and they had weekly COVID testing. So they recorded 4,800 COVID tests between August 2020 and February 2022. Those who tested positive were later asked to complete a survey about their health and behavioral changes. Only 1/3 completed the surveys, and those who did might have been more likely to report lingering health problems, a phenomenon known as non-response bias. Study suffers from two other major problems. First, it doesn't include a control group of students and faculty that weren't infected. The finding that nearly 36% reported long COVID symptoms is meaningless without a sample to determine how common they are among people who never had COVID. Long COVID isn't well defined, but the study defines it so expansively to include problems common among college students: difficulty making decisions, fatigue, anxiety, sadness, trouble sleeping, and the catch-all other symptoms. If a student reported at least one physical or psychological problem, was classified as having long COVID. Physical symptoms like shortness of breath and fatigue can follow non-COVID infections, including the flu. We know this. Some people who get sick with COVID later report brain fog, but mental health problems are prevalent among young people.
And think about the time frame in which this was being done. This was August of 2020 to February of 2022. Raise your hand if you experienced difficulty making decisions, fatigue, anxiety, or sadness during that time period. That's a rough time.
It's a very rough time. So the conclusion is the CDC booster recommendations and George Washington mandates for young, healthy adults are justified in terms of vaccination. She says long COVID deserves more research, but poorly designed studies don't advance scientific understanding, which I would agree with. And, she goes on to talk about some of the other issues. And she says, in conclusion, by exaggerating the incidence of long COVID among young, healthy adults, the CDC deflects attention from mental health problems caused by misconceived pandemic policies. It also fuels public distrust in the scientific enterprise, which may prove to be the pandemic's most destructive long-term effect. And I think that last sentence for me really resonated, right?
Yes.
This is so true. A bad study like this is widely quoted, and it's just a bad study, which you can't conclude anything from, really. So I don't know what you're doing, CDC.
Where did you come up with this, Vincent?
It's in the Wall Street Journal.
Yeah. You read the Wall Street Journal?
No, I don't. Amy sent it to me.
Okay.
Actually, she sent it to me and Daniel talked about it last night on his TWiV update.
Okay.
And had pretty much the same. He said basically, I like emerging infectious diseases, but I have to say I'm highly critical of this paper. I mean, it's a peer-reviewed paper. Why didn't the referees point out all these issues?
Because they're too busy peer reviewing.
They're too busy peer reviewing.
They're too busy
yes.
peer reviewing. Okay, so if you can't read it, that's the basis for that. Alan, I was very impressed that she got this. Okay, it's really
Yeah,
good.
It's cool.
Yeah, that is good, solid science reporting.
Instead of saying a recent study says 36% of students
Yep.
get long COVID, and leave it at that.
Don't just transcribe what the source said. Think about it. Look into it. Yeah.
I mean, you need to know something about a trial, right?
Yeah.
It's not a trial; it's an observational study. All right.
Yeah. You need to know what a clinical study is.
Yeah. Bill sent us a listener pick. This helps me understand TWiV 962, which was with Chiara Mingarelli. General Relativity for Babies. It's a YouTube video. And Bill says, who's going to write Virology for Babies? I'm sure someone already has.
I'm blanking on her name. Our
Angela?
Our Mexican friend who writes the kids' books.
Oh, Susanna
like Paul
Lopez.
Paul has measles. Yeah.
Oh, yeah, that's true.
Wasn't her first book really about virology more generally, or was it measles specifically?
Let's see. I have them posted on my blog, so let's have a look.
I think it was Paul has measles.
Paul has measles was not the first one.
Okay.
Paul has measles virology blog, but there are earlier ones. So there's Paul. Maybe it was Paul has measles. Yeah.
Yeah.
There's then Paul's uncle doesn't want to get vaccinated. Paul and the mosquitoes, and Paul stays home. That was COVID one.
They're great books.
Oh yeah.
Very nice. Yeah.
So that doesn't actually do virology in general, but I'll bet you if you read Paul has measles, you'll get a lot of it.
Yeah. All right. So thanks for that, Bill. And that'll do it for TWiV 987. You can find the show notes at microbe.tv/TWiV. You can send your comments, questions, your picks of the week to TWiV at microbe.tv. And if you enjoy our work at trying to enlighten everyone about virology, please consider supporting us. You can go to microbe.tv/contribute for a number of options, and all of your donations are U.S. federal tax deductible. Rich Conda is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
Alan Dove is at alandove.com and turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.